Good afternoon. I really appreciate the opportunity to share our experience with comprehensive multimodality testing for pediatric leukemias, and to also describe how we use that experience to design most efficient testing algorithms so that we can minimize testing costs but maximize diagnostic yield. And before I start, I would just like to acknowledge all the collaborators and my colleagues from CPM who participated in the study, especially our molecular pathologist, Dr. Himmons, who works with our NGS panel that I will be talking about. So I would like to start with two cases that I think nicely illustrate this idea of the talk, that we should be thinking of, beyond just conventional cytogenetics when we are doing a diagnostic evaluation for pediatric leukemias. So this first patient was diagnosed with AML at an outside institution when he was 19 months of age. And it was megakaryoblast leukemia with a random immunophenotype associated with poor prognosis, and had normal karyotype at the time of diagnosis. Unfortunately, the disease was refractory to treatment. He was still taken to a bone marrow transplant, but relapsed very quickly after transplant. The second case had even more unusual presentation. This case was actually referred to us from an outside institution, and some colleagues in the audience might recognize the case. But this was a 2-year-old girl who presented with a mass in her left mastoid region with no any bone marrow involvement. And actually, the tumor was first thought to be a solid tumor. And differential diagnosis was neuroblastoma or some non-differentiated sarcoma. And the case was actually referred to our sarcoma pathologist as a consult, and he was the one who ordered next-generation sequencing testing. So using our next-generation sequencing panel, in both these cases, we detected the same fusion, which is CBFA2T3 GLIS2 fusion. And what is seen on this slide is a IGV screenshot showing supporting reads that demonstrate the presence of this fusion. Now, this fusion is known to be very specific for AML. It was described several years ago, first in megakaryoblast leukemia in non-Down syndrome patients, but it was later shown not to be just restricted to M7 subtype. It can be seen in other morphologic subtypes. Importantly, it's completely cryptic by cytogenetics because it's caused by a very large inversion of chromosome 16, which does not change morphology of that chromosome, and it's known to be associated with very poor outcome and bad prognosis. So in our first case, this finding explained very aggressive clinical course and would have been helpful with prognosis at the time of diagnosis. But this finding was particularly important in the second case, where actually allowed to make an accurate diagnosis for that patient. And so at the end, the patient was diagnosed with extramedullary AML with no any bone marrow involvement. And she was switched from solid tumor treatment, which was already started, to AML treatment, which was followed by bone marrow transplant, since this fusion is known to be associated with aggressive disease. And I was very glad to hear that she's now doing really well after bone marrow transplant. So these are cases that illustrate the importance of doing additional testing on top of conventional cytogenetics for AML. But the same is suggested from large genomic studies that have recently been published. So these figures are coming from a recently published Target study that showed much higher genetic heterogeneity in pediatric AML than we appreciated previously. It also showed that they're often driven by abnormal gene fusions, many of which are cryptic by conventional cytogenetics, and we haven't been doing excellent job capturing them by using just cytogenetics. And also the study showed that it's completely different genomic profile that we see in pediatric AML as compared to adult AML. So the message would be that we do need additional testing on top of conventional cytogenetics if we want to capture important genetic lesions, and that we probably cannot be using the same assays that are established for adult patients when we are trying to work up pediatric patients. But how about B-ALL, which, as we know, is much more common in pediatric population than AML? So as we know, in the last a decade or so, a large number of new molecular subtypes have been described in B-ALL. And in the figure that's on the left side of the slide, I marked with asterisks those novel molecular subtypes. There are quite a few of them, including PH-like, DUX4 rearranged, MF2D rearranged, DNF384 rearranged. And on the right side are specific aberrations associated with these novel molecular subtypes that are cryptic by conventional cytogenetics. So as you can see, there is a large number of those that we are not capturing by chromosome analysis, and we definitely should be thinking beyond just doing karyotype. So knowing all this, we anticipating that additional testing will be required, our laboratory at CPM at CHLA, we started introducing additional genetic tests to be used in workup of pediatric tumors, including hematologic malignancies. So about 2 years ago, we started using chromosomal microarray analysis, and about 1 year ago, we started using our comprehensive NGS panel. So objective of this talk, or in this study, was to determine what is the clinical utility and increase, if there is any, of a diagnostic yield if we are, when we add these novel testing modalities. So is it really justified using those additional tests in pediatric leukemias? And also, we wanted to use experience from doing comprehensive analysis in a certain number of cases to develop the most efficient testing algorithms for different disease subtypes so that we can maximize diagnostic yield with minimal cost. So for CMA, we are using Affymetrix cytoscan HD array, which I'm sure most of you are familiar with, so I don't need to introduce that platform. And then for next generation sequencing, we're using our custom panel, which we developed in collaboration with Thermo Fisher. And luckily now, even that has been introduced. If you heard the Thermo Fisher talk, vendor showcase earlier today. So I will just very briefly say a few words. But this was custom panel developed in collaboration with Thermo Fisher, which is specifically designed for pediatric malignancies. It has DNA component for sequence abnormalities and RNA component to detect abnormal gene fusions. And it is developed having in mind both solid tumors and hematologic malignancies. In library preparation, we do target amplification, target enrichment using amplification, so that's based on AmpliSeq approach. IronChef is used to increase efficiency in library preparation. Sequencing is done on S5 sequencing instruments using Torrent platform or methodology, and then analysis is done by combination of commercial solutions, so Iron Reporter, as well as some custom solutions that were developed by our bioinformatics team. So we were able to do comprehensive multimodality testing for consecutive cases during a period of 1 year. So between June of 2017 and June of this year, we were able to do cytogenetics, FISH, CMA, and NGS panel testing for consecutive newly diagnosed and relapsed cases of pediatric leukemias. At the end, we ended up having about 100 cases. Of those, 67 were BLL, 23 were AML. And then we had a small number of TLL and MPAL cases that we did not have enough cases to include in this analysis. So looking at BLL, the figure is showing how BLL cases are broken between different subtypes. And the thing that, so this is only presenting classic, well-known subtypes that we have known for decades. And as you can tell immediately, there's a substantial number of cases that does not belong to any of these traditional subtypes like hyperdiploid ET6 RUNX1, MLL rearranged. So you clearly see from the beginning that you need additional workup to accurately classify a substantial proportion of these cases. And then when we applied CMA and NGS, and many of these ended up classifying into some of those novel molecular subtypes like VH-like or CNF384, MEF2D, and others. And then when we started looking at diagnostic yield between different testing methodologies, the first thing that becomes pretty obvious quickly with BLL, that you need technologies that are effectively detecting copy number abnormalities so that you can capture subtypes like hypodiploidy, but also you need good efficiency in detecting abnormal gene fusions, which are other type of common driver in pediatric BLL. So looking at diagnostic yield for key genetic subtypes based on individual techniques, as a single test, FISH panel was actually having the best pickup yield because it's specifically designed to capture common subtypes. So you have probes for hypodiploidy, RUNX1, all those subtypes that are supposed to be common in pediatrics. But when you do combination of techniques, you actually get further increase in diagnostic yield. So if you combine conventional approaches, cytogenetics and FISH, we had a capture of key drivers and key genetic subtypes in about 65% of the cases. But we do see increase when we combine CMA with OncoKid's panel testing up to additional up to 73%. And this was due to detection of those novel molecular subtypes, as we expected, that are not effectively picked up by conventional methods. So those cases that were only captured by CMA and NGS were several cases of PH-like ALL, ZNF384, and even some MLL cases that were caused by unusual rearrangements, insertions, so they were not captured even by FISH. The other strength of these novel methodologies was in detection of secondary cooperating abnormalities. For example, CMA was very effective in detecting small focal fusions that in BALL are very common, and they target genes that are specifically associated with BALL development. And some of those, as we know, are significant prognostically, like Icarus deletions. And then OncoKids was effective in detecting DNA sequence changes. When we look at AML, a single test, OncoKids actually outperformed by far all the other individual tests. It had the highest diagnostic yield for key genetic drivers, and then additional increase was seen when we also had CMA analysis. And then these are, again, cases that were only captured, key drivers were only captured by these novel assays, NGS and CMA. And then, similarly to BALL, the strength of NGS was in ability to detect DNA sequence abnormalities, sequence changes, which are very important in AML pathogenesis and obviously cannot be identified if you only are performing cytogenetic analysis and FISH. So in conclusion, when we looked at the data, we were pleased to conclude that implementation of these additional assays, chromosomal microanalysis and OncoKids, did increase diagnostic yield for primary genetic drivers, key drivers, in both ALL and AML. And it also allowed to detect secondary cooperating abnormalities in these leukemia cases. So we were getting more complete picture and more complete genomic workup for all these leukemias. So based on experience with doing all this comprehensive analysis, this is the diagnostic algorithm that we will be implementing in the future, although it's still work in progress and it might be further tweaked. But we will be starting with cytogenetics and FISH panel because that is still in all practice guidelines, and we also need it for patients who are on clinical trials. And also in the initial workup, CRLF2 flow has been added already by our hematopathology laboratory. And then for cases that are CRLF2 positive, we are only adding CRLF2 and IgH FISH to actually determine which exact CRLF2 abnormality we have in that particular case. We do not routinely recommend doing additional workup, but some oncologists are interested in doing CMA and OncoKids because they're interested in presence of Icarus deletions, which are common in these leukemias, or maybe mutations in JAK1, JAK2, IL-7, which are also common in these CRLF2 positive cases. If we have very typical hyperdiploid, hypodiploid, or ETV6-RUX1, or very characteristic MLL translocation, we do not recommend routinely additional workup because although the additional testing will detect a lot of additional changes, that information is at this time not being used clinically. So we would think that it's sufficient to just have information based on cytogenetics and FISH. For Philadelphia positive cases, we also do not routinely do additional workup, but sometimes clinicians are interested in whether there is Icarus deletion or whether there are mutations that would suggest resistance to tyrosine kinase inhibitors. And then for all other findings, we are continuing with CMA and OncoKids testing. If initial workup suggests AML, so by morphology and flow, it looks like we're dealing with AML. We are again starting with cytogenetics, and then FLT3 ITD testing is routinely a part of a standard workup. If it's suspected APL, we are asked to do STAT-FISH to confirm it, and then there's no additional workup typically required. For typical A21 and inversion 16 and known MLL translocation, we typically don't do additional workup, or we do FISH confirmation so that it can be used for follow-up. And then for all other findings, we're proceeding with CMA and OncoKids analysis. So in the future, what we are looking into is possibility to maybe move CMA and OncoKits into first-line testing, especially for AML, where we have seen much better diagnostic yield with this combination of assays than with doing conventional methods. And that especially will become a case as we are getting better with turnaround times and with costs of these novel methodologies. And finally, for cases that remain uncharacterized, unknown in terms of what is the key genetic driver, after doing all comprehensive testing, we're looking into whole exome sequencing and total RNA-seq. That's now happening on research basis, but we are hoping to move that into clinical testing to be able to fully characterize even those cases that remain unknown at this time. So with that, I would like to conclude and thank you for your attention. And I want to thank my wonderful colleagues at the Center for Personalized Medicine at CHLA.